Now, but here we go. Should be recording now. Awesome.
Perfect. All right. Hello, everybody. So my name is Amalie, and thank you for inviting me to come and talk on EcoMito. This is actually my second time being on EcoMito as the presenter. So this is a really great group. It's fun to be part of International Mitochondria People. So I have actually been on leave from my PhD for a little while because I have just had a baby. So please, if I forget what I'm talking about, I'm going to blame the lack of sleep, but she's a very good baby. So I'm currently off now, but I'm coming back to work in September. So this is a nice chance for me to talk about something to adults about something that's not related to the baby. So for the first chapter of my PhD, I was looking at mitochondrial physiology of torpor and ruby-throated hummingbirds. So thermoregulation is a big challenge for hummingbirds because they are very small. So they have a high surface area to body ratio, meaning they lose heat very quickly. And they also have a high active body temperature, around 40 degrees Celsius, so a couple degrees higher than mammals. And they also, of course, have a very high mass-specific metabolic rate, partly because they use hovering flight. So to support these massive energetic demands, hummingbirds feed on nectar every 20 minutes during the day, or sometimes even more frequently. So nectar is composed of mainly carbohydrates, and they need this fuel to maintain their energetic demand. But at night, they can't see to forage, and so they must rely on fat stored as fuel. But when it gets colder, it's even harder for them to maintain that high body temperature. And they're a migratory species, and they arrive back in London, Ontario, where I live, around May every year. And here is a figure that shows the daily high and low temperature in London, Ontario. Let me see if I can do my uh, okay, I guess I can't do my laser pointer. That's fine. So this is the daily high and low temperature in London, and you can see it can get as low as maybe 6 degrees or even lower when they return. So they have to have a strategy to deal with this. And so hummingbirds use daily torpor to save energy during cold nights and also, interestingly, during migration. So what is torpor? I don't need to explain what mitochondria are to this crowd, but maybe we'll go into a bit of what torpor is. So there are kind of two sides of this story. There's daily torpor and there's hibernation, and both use a state of inactivity characterized by usually low body temperature and low metabolic rate that can be rescued by endogenously produced heat. So this is not a hypothermia, this is on purpose, and they can rescue it themselves by warming up again. So in daily torpor, this is usually short term, hence the word daily, so within 24 hours. And we see about a 70% decrease in whole animal metabolic rate, and body temperature usually decreases moderately between 5 and 15 degrees Celsius. Contrast that with hibernation, where it is long term, so days to weeks. It's usually seasonal, so it only happen in the winter, and you see greater than 90% decrease in whole animal metabolic rate, and body temperature usually decreases quite a bit, so between 20 and 40 degrees Celsius. But this is all data from mammals, so we don't really know how torpor is used in birds as much. The research is just getting started on hummingbirds and other species on how they do this. But in non-volant mammals, which we know mostly about, we also see suppression of metabolic rate on the mitochondrial level. So what we see in the whole animal, we also see mirrored in the mitochondria. So in daily torpor, we see about a 30% decrease in mitochondrial respiration in various tissues, mostly from liver, but also skeletal muscle does it too. So here's a figure that our lab produced many years ago, looking at mice and hamsters when they're torpid and looking at respiration rate. And this was in liver using a carbohydrate-based source, and both of them are decreased about 30%. In a hibernator, we would see about a 70% decrease in mitochondrial respiration in liver, maybe about 50% in the muscle. So you can see here on this figure, at a high assay temperature, 37 degrees, there's a big drop in the torpid mitochondrial respiration rate compared to the torpid ones, or the active ones, sorry. But again, this is all in mammals, so it's pretty well characterized that torpid mammals decrease mitochondrial respiration when to maybe facilitate torpor or during torpor. But we don't know if that is true for birds. So we can say ATP supply is significantly decreased in torpor, but what about hummingbirds? So normally you would see a hummingbird just gone just like that. So that's a cute little video clip I took. But when they're torpid, they look a lot differently. So here's a video that I took of one of my birds in torpor, and you can see that it is not moving. It cannot fly in this state. It's actually actively rewarming right now, so it was shivering. And I'll play the video again. And within about 15 minutes, this bird had gone from a body temperature near 13 all the way up to 40 again. So in hummingbirds specifically, we can see up to a 90% decrease in whole animal metabolic rate. Body temperature usually drops to at least 10 degrees. Also, it can be even lower, as lower than 4 in black metatail. And all hummingbirds seem to do it, so it's pretty widespread. So this is shown in the literature, but we don't know what's happening on a cellular or organelle level. So we came in and we hypothesized that, like mammalian heterotherms, whole animal energetic changes during daily torpor are mirrored on the mitochondrial level in hummingbirds. So just like what we see in mammals, we're going to see this lower respiration rate. So for this paper, we looked at 3 levels: the whole animal level, where we measured body temperature and whole animal metabolic rate using flow-through respirometry, just to confirm what we see in other hummingbirds, but also to relate that to what we're seeing on the organelle level, where we measured mitochondrial respiration using high-resolution respirometry, and on the enzyme level. So we also measured citrate synthase, PDH, and HOAD activity to see if there was differential regulation of those mitochondrial enzymes. Okay, so I'll show you my experimental design. It was pretty cool. So I captured ruby-throated hummingbirds in the summer, last summer and the summer before, and I used this hanging trap where there was a hummingbird feeder inside, and I hid in this deer blind tent with a string attached to the cage. And when a hummingbird came in to feed, I released the string, it closed, and then I caught a hummingbird. So that took a long time, but eventually I was able to get hummingbirds in there and then shut the door and get them into my sample size. So I broke those birds that I collected into 2 groups. One I held at room temperature, and those were considered the active hummingbirds. And then in the other half, I induced torpor by fasting them at sunset and putting them into a chamber set to 10 degrees Celsius, and I just waited for them to go torpid. And then I did whole animal respirometry, where I was able to measure oxygen consumption and CO2 production to determine metabolic rate and to confirm that they were in torpor or not, and get a whole animal metabolic rate like that. And then after that, I took them out and measured their body temperature. So here's the first figure from the paper, which you guys have seen because you read the paper, but I was looking at metabolic rate over time. And so you can see that this individual stayed normothermic for quite some time, about 2.5 or about 2 hours before the rate spontaneously began to drop. Both VO2 and VCO2 decreased significantly, and then it spent an hour or 2 in torpor before the experiment ended. And so I could also calculate respiratory exchange ratio, which is kind of a proxy for RQ. And what this can tell us is what type of fuel they're burning in this case. So RQ was approximately 0.7 throughout the study. It raised a little bit, but it could be just an artifact of that these are so low. It was kind of hard to tell the difference between them. But that indicates that they're burning primarily fat in this state. So the birds were fasted before the start of the experiment, so it's not a surprise that they were burning fat during this whole time. Okay, so then to quantify those data from from a subset of those individuals, I found that there was an over 95% decrease in whole animal metabolic rate looking at VO2, and body temperature also decreased significantly. So none of these results were particularly surprising. We knew that the hummingbirds were capable of doing this, but it was good to confirm in our animals. And I do want to point out that this metabolic rate decrease, 95%, is on par with mammalian hibernators, not just daily heterotherms. So we expected a decrease of, okay, maybe 70%, like what we see in the mammals, but this was even more. So this was like as if the hummingbird was hibernating just within one night, which is pretty fantastic that they're able to do this and not only get down, but they can rewarm very, very quickly in the morning. Okay. The Q10 of this relationship was 2.84, which was sort of surprising considering how cold they got and how much their metabolic rate dropped. But it did make us wonder if this was just a temperature effect. So again, Q10 is the factor by which a rate changes based on a 10-degree change in temperature. And this is a Q10 similar with just a temperature-dependent process. So this could be just the effective temperature, but we wanted to see if there was a more active suppression. So we kept going. Okay, so to summarize this part, on the whole animal level, we saw that hummingbirds decreased their body temperature by about 25 degrees, so to ambient temperature, which was about 13, and their metabolic rate is significantly suppressed, so 95%, and the primary fuel used was fat. All right, so then back to the experimental design. After I completed the whole animal respirometry, I took the hummingbirds out and I sampled their pectoralis muscle, so their flight muscle. And we chose this one because it contributes about a third of the bird's body mass. One of my lab mates says that they're just like flying muscles, like that's really all they are. They're just feathers and a big muscle. And we chose this one because it contributes disproportionately to whole animal metabolic rate and resting metabolic rate. And again, we did the thing that we all do, isolated mitosis, differential centrifugation, and we measured whole animal or high resolution respirometry. Okay, and the protocol that we did was a bit different, so I'll take you through it. So this was with a carbohydrate-based substrate. So we added mitochondria, got state 1, added malate and pyruvate, and then we got what I considered a complex 1 leak state. And then I added ADP, and that brought us to oxphos from just complex 1. And then I added succinate to involve complex 2, and then I considered that oxphos of complex 1 and 2, so kind of total. Inhibited them with oligomycin and considered that leak of complex 1 and 2. And then I did some math to figure out what just complex 2 by itself was. So when we look at the complex 2 results, just keep in mind that, so for example, I would take this rate and subtract this one to see the difference of just succinate. So this is kind of a proxy to measure complex 2 activity. I did not use any inhibitors or anything like that. So this is the way that I did that. And then again, with palmitocarnitine, same thing, but more straightforward. So I added malate and palmitocarnitine, got state 2, added ADP, oxphos, oligomycin, leak. And I also did this at 2 oxygraph temperatures. So I did this at a high temperature, so about 39 degrees Celsius, which is what we've been doing for bird studies in our lab. And then again, I repeated it at 10 degrees Celsius, which is what the chamber for the whole animal metabolic rate was set to. So I did this twice. Okay, and then here are the data. So first, looking at the carbohydrate-based substrates, we did see a difference between normothermia and torpor for oxphos from C1. So there was an interaction effect here, and there was a difference between those two, which is surprising because they're not really using carbohydrates when they're in torpor. So I was sort of curious to see why that there would be a difference here, but it's sort of what we expected. But no difference between in complex 1 oxphos or there was a very not quite significant effect for total. And then looking at leak, we didn't see any changes either. But what we do notice is that, as usual, when you turn down the oxygraph temperature, you see a massive decrease in respiration rate. Again, probably just due to temperature effects, because the Q10 of all these, on average, was about 2.5 or even lower. So I think for some of them, the average was around 2. So again, it's looking like in the cold, mimicking the bird's body temperature, ATP supply is significantly reduced, but when you heat them both up and compare at a common temperature, we're seeing some changes, but not in other substrates or not in other complexes. Okay, so mitochondrial respiration was significantly reduced in cold-bodied torpid birds. But when we looked at the fatty acid-based substrate, the one we were most interested in, because again, birds are using fat when they're fasting and when they're torpid, we saw no changes. So no difference here, which was very surprising when you consider the 95% drop in whole animal metabolic rate does not translate to the mitochondrial level. So there are no differences in the capacity for oxphos when fatty acids are used at a common high temperature, which is the opposite, or at least not what we're seeing with the mammals. Okay, so returning to my summary slide here, we saw no differences in capacity for oxphos with fatty acids, but we can say that mitochondrial respiration is probably significantly reduced in the cold birds. But we wanted to look a little deeper at the enzyme level. So here are the results from our enzyme assays. So we measured citrate synthase activity, and again, we saw no difference. So mito quantity is likely staying constant, which is not a shocker because again, these birds are doing this every single night. So if they were losing mitochondria and making new ones, that would seem like a waste of time. So they're not doing it. But there was also no differences in PDH or HOAD activity. So again, PDH is a rate-limiting enzyme in like the lytic pathway and HOAD in beta oxidation. And we do see some differences in PDH and HOAD activity in mammals and also in other birds. So HOAD in specific, we see increases in shivering goldfinches, which don't use torpor, but they do have a massive thermogenic need in the winter when they're shivering. So we expected that there might be an upregulation in normothermia to keep them warm because they're going to be in this cold chamber. But we did not see that. We wonder if maybe we measured at different phases, like during the arousal, that maybe there would be increases in HOAD activity, but we did not see that. And then just a note on PDH, the hummingbirds were already in a fasted state. So if there were changes in PDH activity, they could have already occurred. So we weren't able to compare fasted versus fed birds because we couldn't get fed birds to go into torpor. They generally don't like to use torpor unless they really need to. So we weren't able to make that comparison, but it's possible that there could be changes there. We also attempted to measure PDH phosphorylation. So we tried to phosphorylate or dephosphorylate the enzyme in vitro and see if that would result in a change in activity. But for some reason, whenever we added the phosphatase, the assay just didn't work and we saw no activity at all. So we weren't able to measure changes in PDH phosphorylation, but it's possible there could be differences there. But since we did see a change in the mitochondrial respiration for carbs, we wanted to see if this was maybe controlled by, again, the PDH activity we saw. So we pooled all our samples, normothermia and torpor are analyzed together in this because, again, we didn't see any differences in PDH activity. And we did see some significant correlations for total OXPHOS and OXPHOS through complex 2. So it could be that PDH is controlling this a little bit with more activity. That means more OXPHOS. But we're not really sure why this interaction was happening because the birds aren't using these pathways when they're in torpor. All right, so returning again to the summary, no differences in PDH or HOAD. So that led us to think, well, why? So we had this hypothesis. We really thought that it was going to be true, but we found that it was not. So the strategy that birds are using to enter torpor or to stay in torpor or to lower their body temperature and metabolic rate is likely different than the strategy that mammals are using, which is really cool. And again, if we look at this figure showing the phylogeny of mammals and birds, what we have here is highlighted are all the birds that are heterotherms. So we have some that do hibernation with an H, and then some that do just daily torpor with a T. And you can see there are representatives in all branches of the mammalian line. So placentals, monotremes, and marsupials all have torpor users. So torpor and heterothermy is likely the ancestral trait. Whereas in birds, we only have one bridge that uses torpor, and there's only one bird that actually is known to hibernate. And so it's likely that this might have been a later adaptation. And so it could be very well that this is happening in a completely different way. And only very few birds, swifts, nightjars, and hummingbirds actually go into deep torpor. Most of them just go into shallow torpor, where their drop body temperature, maybe 5 degrees. But these very large decreases, greater than 20 degrees, are only in a few different birds. So it's likely that they're doing this a different way. How they're doing it, we're not really sure. So that's what I'm going to be looking at in the next couple chapters of my PhD, maybe looking at ATP supply side or just effects of passive cooling because they are so small. So that's all I have for you today. That was in the paper. My email address is here if you want to get in contact with me. And then before we get into some discussion, which I'm looking forward to, I do want to point out that there is a scholarship that I could apply for. So I'm a CGS holder, a scholarship for Canadian graduate students. And there's a travel scholarship involved with this, where if you hold the scholarship that I have, you can apply for this other one to do an international research project, 2 to 6 months. And so I'm kind of in the market for a side project. And so I thought I'd just point that out. Again, I'm not coming back to work until September 2025. So this is probably a 2026 thing that we'd be doing. But I just wanted to put it on your radar that I am going to be reaching out to make some connections for that. But again, we want to focus more on the hummingbirds today. Okay, thank you.
Awesome, thank you. Thank you for your talk. It was great, actually, fantastic. I've learned quite a lot. We'll keep an eye on your on your trip later in the year. If you want to come to
Yeah.
we welcome you happily. Does anyone has any question for Amalie so far? Yeah, Nico, I've seen your
Hey, Amalie, thanks for the talk. Yeah, I will gladly recruit you, but I'm in Canada, so it's not working
Yes, I
apparently.
know.
Yeah.
No.
All right, so you pretty much pinpoint the PDH as probably the kind of the culprit there. Okay. I'm
Hmm.
just thinking, uh, did you try to find if it's not PDH, but maybe upstream PDH? So, for example, pyruvate transport inside the mitochondria, or even some kind of post-translational modification in terms of pyruvate kinase or stuff
Hmm.
like that, that's kind of impair the pyruvate oxidation.
Yeah, no, it's a great point. So we do think that there's a relationship there, of course, like the correlations are pretty clear, but no, we did not look at upstream in the glycolytic pathways or at transporters. It would have been really nice to see if PDH phosphorylation state was changing in another way. So we kind of just tried to do it in vitro in the assay, and then when that didn't work, we were like, okay, we're running out of time. It's also hard because the samples we get from the hummingbirds are just so small that we don't have a
Mhm.
lot of capacity for troubleshooting. So if we do one
Yeah,
assay and it doesn't work, we kind of run out of PEC, and then we've got to wait for a whole other summer to collect more. So it would have been interesting to do those things, but we tried to narrow down our focus to get as much done as we could with what we
Yeah,
had.
and I guess that antibodies are limited for
Yes.
reactivity hummingbird as well.
Yep.
Okay,
Yep.
thanks.
Thank you. Glad you made it.
Got a quick follow-up question on this one, actually. Did you manage to do metabolomics or GCMS, and you could look at what substrates or products
Yeah, that's a
accumulate?
good idea. So we didn't do it, but I know there's a lab at our university who I'm friends with, the lab mates, and they have a GC and they've done other things. We did in another side project, we did some metabolomics on hummingbird brains, but the results didn't make a lot of sense. Like no one really knew what to make of it. So we are working on analyzing those data and figuring out what's going on. But again, it was in the brain. So it was part of a different brain study. The brains are usually not turned down very much in torpid animals, so that goes for mammals and probably birds too. So brain activity doesn't really, like the brain activity decreases, but it's kind of the mitochondrial activity really doesn't in the brain. So I don't know, but it would be really cool to do that.
Okay. So Tony, I think you got a question, and then Emma, you can follow up after.
yeah. Great talk. I loved it. I've always been fascinated by hummingbirds. Such a cool model. Hey, it's just one of the things I might have this wrong, but birds don't have a UCP like system for warming up. So they
Yeah.
use sarcolipin. My understanding is so that they have an uncoupling of the SR. Have you thought that it could be regulation outside the mitochondria? So like just a complete suppression of ATP turnover, and then
Yes.
they can just turn that on and off.
Yeah, so that's actually chapter 2 of my PhD. So I measured
Ah,
the lipin abundance and circa activity. And so I just finished that right before I went on mat leave. So I have all the data, but I haven't quite finished the analysis yet or kind of put that on pause. But yes, 100%. It could be like the demand side. So yes, birds don't have brown adipose tissue and they don't use UCP1. There's an avian UCP, AVUCP, but it's
yeah,
not clear what
transporter.
exactly.
Yeah,
Yes, yeah. So it's not clear if
yeah.
it's doing the same thing, probably not. But yes, I'm going to be looking at the demand side and circa, circolipin, all those things, chapter 2.
So you've done it, have you?
I've done it, but I haven't analyzed it yet. So yes.
Okay. We've got some ideas, but it's probably too late. Anyway,
Well, maybe we can maybe we can do it. I don't know. It might be too late, but I'd
yeah.
love to hear the ideas. We can chat later.
No hummingbirds down here, unfortunately. Mm,
Yeah.
okay.
Yeah.
Thanks very much. That was great.
Thank you.
Emma, I think you had a question as well.
Yeah, it's great to get an update about this work. It was a great presentation, Amelia.
Thank you.
So I have a quick question. Did you have any birds that didn't go into torpor?
Good question. So at the beginning, we didn't fast them at sunset. So I fasted them 2 hours before sunset. Before, when we just started this, They could feed as much as they want. And then as soon as sunset happened, I threw them in the chamber and they did not want to go into torpor. So it seemed like because they were in captivity, they had lots of nectar. They had no one to compete for it. They could just really load up and they were very fat at night. So they didn't use torpor. But when I fasted them for 2 hours at sunset, every single one did. So I've read that, so female hummingbirds, when they're incubating their eggs, will avoid torpor because they want to keep the eggs at a more constant temperature. They will go into it if they're extremely stressed. So if they're very fasted or if it gets super cold, they will have to do it. But as far as I know, the hummingbirds, yeah. So in general, with most heterotherms, torpor seems to be not good. So they usually want to avoid it. So that goes for like squirrels, for example. If they can store up a lot of fat in their bodies, they're going to try and avoid torpor as much as possible, but they will eventually have to use it. So I think the same goes for the hummingbirds. But no, I was able to get them all to do it if they were significantly persuaded by no food and cold.
If I'm remembering correctly, the adipostat paper that Chris, your lab put out, they actually showed, wasn't there a benefit that like birds migrating won't use torpor, ruby-throateds won't use torpor to prevent like
Yes.
losing all fat stores?
Yes. So that's called the migration assist or torpor assisted migration. And so hummingbirds and bats will both do this. And so, yes, so I was looking in the summer outside the migratory period, but during migration, hummingbirds will use torpor much more often, especially even if they have enough fat. So usually in general in the summer, if they have enough fat, no torpor. But in migration, it switches and they start to use torpor more, presumably to save that energy for a different thing to migrate. So yeah, that's right. Eric's paper did that. And actually, I have to credit him for the hummingbird trap idea. So he was the one who kind of invented that hummingbird trap door thing. But yes, during migration, it switches and they'll go into torpor even if they're fat, which is really cool.
Yeah, no, that's awesome because not to take up too much of your time, but people always ask me because I ban hummingbirds too, and they ask me, well, when do they use torpor? And I now, I used to couldn't answer that other than we know that it's like energy saving, so probably colder days, but they're like, oh, do they use it in breeding? Do they use it during migration? So no, thank you. And we actually use a very similar trap
Oh,
that
nice.
someone made in the 80s and 90s, some of our older banning mentors. But yeah, so I'll have to send you a picture of what our traps look like at some
I'd
point.
love to see that. Yes.
Yeah,
Yeah.
and
Oh yeah, thanks. I would love to see your traps. I struggled with mine too because sometimes the door would close, but the hummingbirds are fast, so the door would be falling through the air, and then the hummingbird would launch out. And so I got this magnet system where there was a magnet on the top and then on the cage, and then that snapped it shut, and they couldn't escape because they're sneaky. But yeah,
I'll
that's
get
really
with you
cool.
about that. I have some recommendations because yeah, I've caught many a hummingbird. So, but very cool.
Yes.
Thank you.
Okay, thank you. Yeah, I'll definitely like to have that advice because they're sneaky. Yes.
Seems to be
Oh,
easy.
and sorry, I just thought of one more thing. When people ask you when they use torpor. So yes, increases in migration. What we really wanted to do, a study that we probably won't do now, but we thought would be cool is to look on the wintering grounds when they're in the tropics, like go down to Central America and Mexico and catch the same species, ruby throats, and see if they're doing torpor in the winter. We thought that would be really cool. A paper just come out looking at tropical species, and all those hummingbirds also went into torpor. So it's likely that they're doing this way more often than we expect them to.
I've got one more, Jules.
Yep, yeah, go for it.
I just noticed in your trace where you had your whole animal respiration where they're going into torpor, they've got a lot more spikes in activity before they go in. How does that relate to, say, squirrels and things? Is it like a preconditioning thing, do you
Do you
think?
mean like the test drops, like that sort of thing? Here.
Yeah,
Oh,
yeah.
I don't know if I can, for some reason I'm trying to go back, but it's not working. Here we go. So like this
Getting pretty
part
glitchy
here.
down here in New Zealand with the feed.
Yes. Okay. Can you see my cursor?
Yes.
Yeah,
we can.
yep. So
Yeah, okay.
it's
Yeah, so
slide
do you
8.
mean just
Slide
the
8.
spikes all over here or just right here?
Yeah, there we are. There was a bit of a lag.
Oh, yeah.
Yeah, I mean you've got a lot of. Oh, they're starting to drop though, aren't they? So
Yeah, I got pretty excited actually when I saw them starting to drop and then it would spike up again and then drop again and then spike up because in hibernators we see these test drops and that's usually in the days to weeks before hibernation is their body temperature will start to drop and then go back up. And so we think that yeah, that's sort of no one really knows, but it's kind of like readying their bodies to go into
yeah,
a deeper torpor. So this all happened within a span of maybe like 10 minutes. So I don't know if that's enough time to do the same thing. I searched a lot of mammal papers, a lot of mice papers to see if test drops happen in daily torpor in mammals. So if at night the mice will start to do test drops. And I found one case where someone said test drops in a paper, but they said that this is real torpor, not a test drop. And so it's very hard to know if daily heterotherms use them or not. It might, I don't know. I showed it to my supervisors and I got excited about the test drops here, but they said it's probably just an artifact because it does look very spiky over here as well. And
yeah,
so
yeah.
yeah, hard to say. I think I need to change the flow rate to get a bit of a higher resolution here, but I don't know. It was exciting though.
Mm.
Yeah, because it does smooth out a lot
It could
once
be
they go.
interesting to look at what's going on in terms of yeah, but it could be interesting to look at what's going on in terms of antioxidant release and things as well. Birds are chock-a-block with uric acid, and it'd be interesting to see what they do compared to a mammal.
Yeah.
So, and they've got all sorts of crazy things in their genomes to do with their antioxidant response elements, and yeah, cool. That was excellent.
Really cool. Thank you.
Anyone has any other question? I might have a quick one actually, if no one has one. You know if they also use alternative substrates, like instead of just lipid and fat, they're also using something else. Like Tony forwarded me a paper like 2 weeks ago or something. They might
Right.
use ketones. And we know that sharks do use ketone as well. They've got really
Yeah.
odd metabolism. And so do you know whether they could actually use alternative pathways and maybe shut down other pathways?
That's a great question, and I have no idea. So it would be cool to look at that, but yeah, we didn't do that. So there were a lot of different things we could have done, but we had to prioritize again based on how much tissue we collected. But yeah, I don't know. It could be because they're not using this one or they're not shutting down this one. So they could be using other ones, shutting down in other places. Yeah.
How much tissue do you get actually from the pectoral muscle? Like just roughly.
Uh, let's see if I remember. So they're kind of like this size, but
Okay.
then so I had to portion them. I can't remember. Maybe it was 0.04 grams, something like that. I was sampling them 2 summers ago, but I had to split them because the stuff I used for the oxygraph I couldn't use for the enzyme assay. So I was already
Yeah.
like, yeah, especially
Yeah.
because this study we did it all in the same animals, every level. So yeah. And now they're gone. I'm fresh out. None left.
Oh, there we go. So beta hydroxybutyrate, is it?
Yeah. Mm.
Yeah.
Yeah. Yeah. Pretty much all I mean, at this point, I don't think we've tested them on all, but just like a lot of people who are not actively collecting and during migration, you can at least get blood sample and look at BOH. And pretty much at this point, it's pretty established that during periods of fasting, so active migration, BOH goes up at least in plasma.
Hmm.
It's something that we're sort of often we neglect these things, and I know that in a mammalian system that we ourselves are very good at using ketones, particularly the heart. So it would be kind of cool to look at,
Yeah.
to look at ketone oxidation. Yeah.
Mhm.
The one thing that if they are using a lot of it is what do they do with acetone?
Mm.
So
I'm
cool.
writing it down.
Breakfast is waiting.
We'll have to wait a little bit more. Anyone has any other question for Amalie? If not, well, I think we can wrap up the session. I don't know what you think, Lisa.
Yeah, and thank you very much, Amalie. The talk was super interesting, and I really
Yeah.
hope that you will find a way to explain what happens with these hummingbirds and how they can do that, because it's just amazing. They are amazing birds.
I agree. Thank you very much.
Yeah, thank you
That was awesome.
for your time. Congrats again for being
Thank you.
ma'am.
Congrats.
Enjoy those days because they don't last really long.
I know.
Nice.
Yeah.
So yeah, have a great one, and thanks everyone for joining. It was beautifully presented, as people say in the chat. And we'll keep in touch for the next journal club next month.
Yeah, it should be before the 20th of December, but you should receive an email very soon about this one, so no worries. It will arrive soon.
And with the link.
Perfect.
Thanks everyone.
Stay at work,
Thank you.
Thank you, everyone.
See you.
Bye.
Bye.